Hello, in this video we will be looking at the eukaryotic cell cycle. In the eukaryotic cell cycle, a cell begins by entering into G1 or GAP1 phase. As the cell begins into this phase, there is a decision that is made as to whether it will go into the resting phase and not basically do anything and just take a rest for a while, or whether it will proceed through the full cell cycle. But it's important to note that the very first checkpoint is in the GAP1 phase. Let's take a look at it now. So at the G1 checkpoint, the proteins that are involved will be taking a look to see, number one, if the cell is ready to go into the cell cycle, which means it needs to grow in size and you need the duplication of the organelles. If everything is okay, it will proceed through the cell cycle. Let's see what this looks like. Take note that from the point of starting into the cell cycle until it reaches the end of GAP1, the cell has 2n number of chromosomes. This is called the diploid number of chromosomes and is the normal number of chromosomes that the cell contains. A human cell contains, of course, 46 chromosomes. Next, the cell will enter the S or synthesis phase. Many people do not realize that the synthesis phase of the cell cycle is actually DNA replication with DNA polymerase. Let's take a look at this one now. Note that during the synthesis phase, the DNA has doubled. So effectively, all the DNA in the cell, the 3.1 billion base pairs, have become 6.2 billion base pairs. And now the cell cycle can continue. Now the cell cycle will enter into GAP2 or G2 phase. There will be another checkpoint here to make sure that the DNA has indeed been replicated and that the cell is large enough and has enough organelles to undergo mitosis. At the G2 checkpoint, there is a check for whether or not the DNA has been replicated. And as I said, is it big enough? Does it have enough organelles to undergo mitosis? It's during mitosis in which the entire process of dividing up the cell, including the chromosomes in the cell, because we have basically two diploid amounts of DNA in the nucleus and they have to be separated. This is what is going to happen during mitosis. So this G2 checkpoint is very important. Now let's enter into mitosis. Okay, mitosis has 4 different phases. You've probably studied mitosis more than any other part of the cell cycle. In fact, most people do not even realize that it's actually during the S phase or synthesis phase that the DNA is replicated. In mitosis, we have prophase, we have metaphase, we have telophase, and then we have anaphase, followed by cytokinesis, when you actually get the division of the cell membrane, nuclear membrane and cell membrane, such that you have 2 identical daughter cells. There is a checkpoint in mitosis, and one of those checkpoints is one that checks to see if all of the chromosomes are actually lined up on the equatorial plate within the cell. Note that at the end of mitosis, we have 2 cells. Each cell has a diploid number of chromosomes, which is the correct number. And at this point in time, then, the cell membrane will close off and we will have completely 2 different identical daughter cells if everything went correctly. And then the process begins again. I hope that this video was helpful in clarifying the different parts of the cell cycle, G1, G0, synthesis, G2, and mitosis. And that now you realize that in mitosis, this prophase, metaphase, telophase, and anaphase, which you have been studying in much of your scientific career so far, actually is a very small part of the entire cell cycle. Thank you.